just kind of discuss a bit of the sampling, handling, storage, and shipment guidance for classification. It's one of the shorter things, although we lost 15 minutes, so I'll try and finish up. This was one of the topics that I was asked, excuse me, to address, because presumably certain monitoring programs, these are some of the nitty-gritty issues that you need to consider. Okay, goals of sampling. Obviously, I think we'd all agree that we want to obtain a representative example of a water bloom event that can be used for identification and enumeration, maybe cyanotoxin analysis, and eventually some sort of guidance and what do we do? Postings or whatever you do. Want to use that information obtained to make decisions regarding potential or observed harmful effects from the presence of the toxigenic cyanobacteria. And a final point would be that, again, keeping in mind, not get the cart before the horse, sampling and analysis should be based on project goals, not goals based on sampling and analysis. Don't go out and collect and then decide what you're going to do. Otherwise, you probably wasted some time, effort, and presumably your budget. As an example, in Oregon, they had a HAB program that was funded from this 5-year cooperative agreement with CDC. They were one of the 11 states in that program. California was not. Their program overview was coordinating response to HAB events, alerting public when a toxic bloom is detected, tracking occurrence and characteristics of the bloom, investigating reports of HAB-related human and animal illnesses, maintaining relationship with the partners, ensuring continued quality environmental health data, and raising awareness through education. Those are logical things that you would have as goals. Now, in terms of setting those, some of the key references that states have used, and I always cite in getting that kind of background information. Obviously, this book has a very nice chapter because it was one of the things that we paid a lot of attention to. 12, determination of cyanobacteria in the laboratory. And I've actually duplicated that chapter in the handout manual that you have, the little blue, or the other one, the one that's in the binder. That chapter 12 is duplicated in the back for you, which we'll talk about after lunch. Chapter 2, sampling and identification, the methods and strategies in the AWWA manual. That's this one. I heard somebody say, oh, I have that. This manual was fairly new and was designed specifically for algae, and there are at least 2 or 3 chapters in here that target the cyanobacteria as well. In fact, the dichotomous key that we'll use this afternoon is in this book as well. And again, if you're members of AWWA, I think you all got a copy because you pay a lot of money for membership. But if you have to buy one, they're a couple $100. But it is and does cover most of the issues with regards to algae and does focus a bit on cyanobacteria. The new one that's coming out is going to have even another chapter, will carry significant revisions for sampling, handling, identification, cyanobacteria. And this is by a woman named Ann St. Amand, who runs Phycotech in Wisconsin, that does a lot of the identifications and things nationally. Maybe some of your groups in California send their samples. I know Oregon and Washington often send samples all the way to Wisconsin for identification. The standard methods, sorry, got ahead of myself. The standard methods, which is the big Bible, the one for water and wastewater, the big one. The volume I have is 2005, but there may be another one out. So that's the one that's going to have this revisions for including cyanobacteria. So right now the standard methods book does not have specifically information on cyanobacteria. That's the one that's going to carry her information. This, not this. This is out. It's probably not going to be revised. So those are 3 places, and I've actually used things out of there in the next couple of lectures that we'll do. So sampling considerations: location and use of the water body, whether it's a drinking water, recreational, stock pond kind of thing. Goals, sampling efforts, identification, enumeration. Maybe part of our program is chlorophylls. We'll see why. Chlorophylls is a surrogate for biomass. There are other ways to do biomass if you have to collect biomass data. And then also for cyanotoxin testing. So typical locations that I've seen used here, there, and everywhere for blooms in thermally stratified shallow lakes and reservoirs. You know, if it's recreational or if you're sent out to get a sample, usually you go out and look for the scum. So obviously shoreline scums. Planktonic scums on the open water are often more representative of what's in the lake, but that means you've got to have a boat. 3, phytoplankton scums in or on lake sediments often are important, maybe not so important for swimming, but if you can't see the scum, there certainly are blooms on the bottom, and they can be toxin producers, the benthic. Dispersed populations of phytoplankton within the epilimnion being the top layer before the thermal stratification. Homogeneous populations of phytoplankton dispersed throughout the water column during well-mixed conditions, and scums under the ice. As I mentioned, some of these blooms can actually overwinter, and I hadn't realized this myself, but working in Scandinavia, which has a lot of ice, you'll get red ice, and that red ice is cyanobacteria, literally frozen in the ice, thawed out, and it grows. And they can be produced toxins. Subsurface or metalimnenic phytoplankton maximum. So these, you know, if you were set out to say, okay, where do I collect? These would be the locations. Now this would be if you're just monitoring a lake or a reservoir. If you were focused on drinking water supplies, then it might be a little different. In drinking water supplies, the studies that I did, one was the AWWA study that I did in the 90s. We had 50 utilities. Several of the big ones here in California were involved: Metropolitan, East Bay Mud, and a couple of others too. I had them sampling surface, the middle of the water supply body. I wanted to know what was in the water in sort of a more general sense. Then I had them take intake and mid-process stream and finished water. Mid-process stream because, you know, you come in, you do your sedimentation, you do your, you know, maybe your carbon treatment. You need to kind of have a sense of, is that initial process working, especially if you don't have you know, advanced oxidation in the system. And then, of course, ultimately the finished water. So it sort of makes sense, but these were the 4 sample locations that these utilities used and sent me samples on a regular basis for 2 years that went into that study. And then recreational, and then stock water supplies. Recreational, obviously you probably want to go to where the activity is, but it's pretty important at that active area that you go under the surface, because lots of blooms aren't on the surface. They could be under the surface and still in that recreational area. And then, of course, if you have jet skiers and jet boats and water skiers, then you need surface of the open water where especially the spray. Spray is a source of exposure, a significant source of exposure, inhalation, and contact with the face as well. So, on the St. Johns River, this is what it looked like when we were sampling. This is in Florida. If you're doing your monitoring things, it's the usual kinds of monitoring efforts. Sample locations, or sample collection. I never worried about gloves, but I know that when you're an agency and you've got all these other things to consider, then I definitely recommend using gloves, at least. The question becomes, do we also use a mask? And of course, I don't think it's necessary, but you know, it depends upon who you have sampling, and they say, I'm not going to sample unless you give me a mask, so then give them a mask. But usually what you'll do if you go into a public recreational area and you're wearing gloves and a mask, it's like everybody's going to go home, so, or at least you're going to make the local newspaper. But anyway, you know, collecting and filtering and processing your sample. Unless you've got a graduate student that you would not like to have any longer, probably immersion is not recommended. But anyway, some of the references here where I get this kind of information. This is the study I did for AWWA in 2001, assessment of blue-green algal toxins in raw and finished drinking waters. The EU, European Union, spent an awful lot of money bringing countries together in Europe and have done an awful lot of work, a lot more than is done here in the U.S. And they've done a lot of publications. One of the more significant ones is 2008 that came out in this EPA book, and that's entitled Cyanotoxins Sampling, Handling, Processing, and Toxin Uptakes. So there are those references. And even a place like the North American Lake Management Society has a website where they tell you how to sample. So there's a lot of information out there, and you know, I highly recommend you go to those places, get that, sit down, and set your own policies. Well, some of the other things that I think are more practical, people ask, and it's a legitimate question, what kind of containers do I use? You know, the safest one is to use, you know, sterile or disinfected or at least clean glass, amber or clear. I don't see any distinction where amber, you know, often if you're a health authority and you're collecting for health, you know, health, they use amber glass bottles as the standard thing, and that's fine. Generally, you're processing this material soon enough or storing it so that the amber is not going to be that protective of decomposition, so clear is fine. The other issue is, do you send people out with glass into the field where it can break? And that's the other consideration. I usually don't, so that plastic is recommended. There are some studies that show that you might lose a bit, toxin absorbed, but that's only in very special situations where the toxin is being put into ampules as standard reference materials for an analysis kit. If you're just collecting water and it's going into plastic, you're not going to lose that much. So I prefer high-grade Nalgene plastic. And again, you don't have to pay extra for amber, you can get the clear. And now, the, you know, other practical things is, well, do I collect and then can I reuse it? Sure, you can reuse it. I would, you know, clean them with some, you know, a good lab-grade detergent that's environmentally friendly, and then a rinse with dilute Clorox solution just for slight disinfection, and then reuse them over and over and over again. Recreational water is the same as drinking water, and livestock same. So I guess line is, this might be a kit that you have. In fact, these are a picture of the exact kits that we sent 50 utilities for the AWWA study. Had 4 bottles for those 4 locations, and we had instructions and bags and samples and gloves and everything was in there. And in fact, for this AWWA study, we had them filter. And so we had actually, because one of the things we wanted to find out was, do you need to, you know, what kind of losses do you get if you filter the sample and process the filtered sample back in the lab? So we had them collect unfiltered, and then we had them filter and ship us the filter papers. We used filters that we could clear and actually analyze directly for toxins. So that was what was done back then. So out of that utility study in 1990 to 91, we were collecting 4 samples. Sample 1 was surface, subsurface, intake, influent, and effluent. And this literally was the instruction manual that was in that kit, just to give you an idea of how we did those things at that time. So here, see, we filtered, send the filtrate and the filter, because we also tested the filtrate to see whether we lost toxin into the filter or filtrate, or if we got most of it on the filter. And in these cases, we did conclude that you could use filtered material and only process those filter papers. If you're doing, now we're talking toxin testing. We're not talking identifications, but for toxin testing. We didn't do identifications for this study. Other things that are a bit of consideration, most utilities, when they place their intakes, they have the ability to withdraw water at different levels. And primarily that's part of it, because sometimes in the wintertime, if they are below, down in the anoxic zone, then they've got more concerns about taste and odor. So they'll bring their intakes up. Likewise, if there's a bloom down here, then you can move your intakes up or down to get around a bloom. Often their surface intakes aren't an issue at all, but it does allow you to adjust that. Most utilities have that, although I have seen utilities that don't. They have one intake and that's it. And if they can't withdraw from that intake, they can't use that water. So adjustable intake is valuable to avoiding blooms at variable depth. And keep in mind that blooms do go up and down day and night. So there's a diurnal fluctuation of the bloom. Not always, especially in these heavy scums, it's all up at the top, but blooms do tend to move up and down. That happened. Okay, conventional treatment plants will want to keep those cells intact. That's the other take-home point. The biggest problems I've ever seen with utilities and toxins is the fact that in initial processing, before they remove the cells, they do something to stress the cells that dumps the toxin, and then they've got soluble toxins that become more of an issue than just removing cells. Just keep in mind, along with everything else, cells get removed in those first stages, whether it's sand filtration or flocculation and sand filtration. So they want to be able to get as many cells out as they can, because that's where most of the toxin is. So the US EPA sampling recommendation, I throw this in because this is the new one. This is June 2015, one of those documents the EPA just put out. Samples should be handled properly. A lot of this is just your usual proactive jargon, but public water system should sample collection handling procedures established by the method and laboratory performing the analysis. The thing that seems to come out of this is that go to the lab that you're sending them to and have them tell you what to do. So that's what this basically says. And of course, when we don't tell you anything, then go down here and get guidelines or something. But this is what is the official EPA, US EPA statement at this point.
So they seem to suggest amber glass containers.
Exactly, and they have been convinced that amber makes a difference, and I'm not. But again, if you're setting guidances, you often go to the maximum. And of course, if something goes wrong and you go to them and they'll say, well, you didn't do an amber, I can't help you. So, I guess in that sense you need to use amber. I honestly don't think it makes any difference. But if that's what's recommended and that's your policy, then obviously do that. They were heavily influenced by, and the reason for this is digression, but it shows you what happens. They were influenced by what happened in Lake Erie last year. And one of the problems at Lake Erie is they rushed and they grabbed samples and they sent them to half a dozen labs, and they sent them in different ways, in different forms. Every one of those labs did something different, and they all got back all these conflicting results. And when they all said, then they decided, well, we have to be consistent, and then when it comes to being consistent, what do? We go to the extreme, and the extreme was, I'll just use amber glass. Because they didn't know, why did we get these inconsistent results? And it often was how the lab was actually running the test. They all had the same test, like an immunoassay, but they all processed the samples differently. So that's why these things get in there when maybe they aren't needed, but they're there.
So they also say samples generally should be analyzed within 5 days, but is that critical, do you think?
Well, it's critical in the sense that the utility is sitting there wanting an answer yesterday.
But in terms of the answer that you get,
yeah.
Does it matter? Like, I mean,
No,
some
five
some
days.
things you have to make sure they get done within holding time. Some things,
Right.
The holding time. Is
No,
not
the processing time is not critical, but what's critical is getting the information. 5 days, because that's a working week, But no,
But it wouldn't compromise.
I could tell you at the end of 5 days, you're still going to be able to run that sample the next week. And unless you left it at 110 degrees on a loading dock or something, but if it's in the lab in a cooler, then no. I have samples that are 50 years old and still as active as they were, but they were dried and put in a freezer too. But the normal times at which you need this kind of information, 5 days isn't the critical. But everyone wants to know, what do you recommend? Okay, this is what we recommend. And generally speaking, if you say ASAP, then it's next year or something. If you've worked in your agencies, you get off to other things. You have to set some criteria. 5 days is reasonable. Yep.
I may have missed this, but did you say something about refrigeration or cooling?
Well, normally refrigeration, because if you're collecting raw samples,
Sorry.
if you're collecting raw samples, you do have other, cyanobacteria could be active if you don't refrigerate. Just the normal kind of holding procedure that you'd use for any kind of water sampling. Yeah.
You mentioned any kind of preservative sample volumes?
Yes, I will do that. We're going to get to that, sure. So just hold that thought. Okay, handling of samples. Collections made with clean containers, amber or clear, using gloves. The recommended preservative is Lugol's, and I'll actually expand on that a little bit too, because questions are, what about formaldehyde? What about glutaraldehyde? Well, really it comes down to what's safest for people to be handling, and Lugol's is. People familiar with Lugol's? It's an iodine-based solution, and it's usually you have a stock solution of Lugol's and you use 1 ml and 100 mils, or the rule of thumb is a light tea color to the final sample is sufficient for preservation of all the algae that are there. If you're doing cyanotoxin analysis, however, no preservatives are recommended because they will interfere with the test, especially the immunoassay test. Okay. Obviously, if you're handling the cells, dried cells, for whatever reason, they should be in a controlled biohazard hood. Dried cells are very carry a high static charge, and they'll just float everywhere. Storage of samples. A little more on your question about preservatives. There's really no perfect preservative. Formaldehyde, is what we used to pickle pigs in for intro biology labs, which we don't do anymore. But it has its own hazardous, its fume, and the other things with these very strong oxidative compounds like formaldehyde or glutaraldehyde, they tend to distort the cells. So if you're doing biovolumes or cell size or anything like that, then you have troubles. if you do use it, it's 2 to 5%. I mean, some agencies, especially health agencies, where that's built into the system, they will use formaldehyde because they use it for tissues. But Lugol's is probably the most innocuous. But there's even a little bit of loss. For example, cells will lose flagella. There's a color change over time, but again, if you're working these up within a reasonable length of time, and cyanobacteria don't have flagella or cilia or things, so that's not an issue. So Lugol's is really the best. Glutaraldehyde is probably the best preservative, but it's also the most toxic, and so most agencies don't want to get involved with that at all. Shipment of samples, shipping in a cooler box that contains cold packages. Express overnight is generally best, doesn't have to be, but that cooler box is often recommended so you minimize spillage and you don't know where it's sitting in the interim. So I always have my samples shipped express mail in a cold box, or at least in a box that's packed with a cold pack in it. If you are working with the purified toxins for whatever reason, and I suspect many of you won't be unless you're just buying them, they're okay in the test kits. But if you're sending other significant amounts, they more recently, especially since they kind of get tagged as biological warfare kind of things, they're considered hazardous, and DOT, especially microcystin, is on the DOT list for restrictions. And so some places like FedEx won't accept microcystin for international shipment. Within the country, it's fine. So in general, this is not an issue, but it is if you're particularly doing something. We always had to deal with it because we set reference standards here, there, and everywhere. So out of an Ohio workshop I did 2011, this is what Ohio kind of summarized. They recommended 100 to 500 mil samples as your standard sample size. Part of the reason for that is if you're, especially a finished water or a very processed water sample, you need at least 500 mils to be able to detect toxin in it at the level of the 1 part per billion guideline. If you're just doing algae counts and algae identification, 100 mils is plenty. So that's what that range was chosen for. They said always collect your samples some distance from the shore and about 20 to 30 centimeters. In other words, if you're only collecting one sample, this is the one that's going to give you the best representative of what's there. Now, if you can collect and afford to collect multiple samples in locations, shoreline, Center Lake, other places, then that's okay. But if you're only doing the one, this would be the one best one to collect that. Concentrated bloom is okay for toxin analysis, but in terms of setting some sort of guidance for hosting the lake, then you need a more homogeneous sample. But if you're doing a toxin test, just grab anything. The thicker, the better. Although, if you have to sort of, the kickback will be that people will say, well, that's not the toxin level that's out there in the middle of the lake. That's true. But you're at least getting a measure of the toxin in a gram of cells. And then you may have to have other information that tells you, well, what are my cell counts at other locations. Wear gloves, maybe a mask. Keep samples cool, shade in the dark. Lugol's, and if you can't use Lugol's, glutaraldehyde. Yes?
Is the 500 milliliter sample size for ELISA analysis, or would that be for LCMS kind of things too?
Good question. How am I going to answer that? Well, the 500 mils is going to give you a volume. Even if you have low cell counts, you'll still get enough to do a test. You're probably going to process that whole sample. You're either going to filter it and take all the cells out of it. You're going to do a sedimentation and take all the cells out of it, or you're going to bulk freeze dry that whole sample. If you were sending a sample to a lab for testing and you sent them 500 mils, they would process that entire 500 mils, and then they would have a biomass, 100 milligrams, a gram, or whatever. They would take a subsample of that, give the toxin analysis, and then they would back calculate it to the biomass that was there, and back calculate that to the volume that was there, and tell you how many micrograms of toxin in a liter of water. And to be able to say that you are detecting the toxin, if you only sent them 100 milliliters, that might not be enough to get onto the sensitivity of the test if it's low levels, and then you'd miss a positive and you'd have a false negative.
Yeah.
That makes sense?
Yeah.
So 500 mils is kind of the magic number.
Okay.
And I used to say a liter, but often a liter is just too bulky and too heavy, and over time we've kind of concluded that 500 mils was good enough too. Even if I collected the 500 mils of finished water like Toledo, we'd have enough toxin in there to come up with a number that would fall onto the detection level for the kit.
Okay.
Does that answer the question?
Yeah, I'm worried because our lab is just switched to 250 milliliter bottles, and we're sending whole water samples, so it's the particulate and the bulk all together.
Right.
For ELISA, so I just wanted to make sure that we're giving them enough.
Well, most likely.
Yeah.
Most likely. But 500 is a little more protective of getting enough. I mean, it's going to collect, you're going to, most of the time. But if you're really down at that detection level, if your guideline is 1, probably 250, if there isn't much there, wouldn't be enough. Yeah, that would be, but the answer would be most of the time it's going to be okay. Okay. Yes, that's it. More questions, sorry.
It sounds like you're collecting the organisms and then you're measuring the toxins that are in the organisms, as opposed to what if the organisms have died in the water and then you have toxins.
Good
in the
question.
water. So are you're just measuring what's in the organisms?
No, because we just discussed, we take that 500 mils and we process the whole thing. So you're getting everything that's in there.
Okay, so you're concentrating the toxins.
Well,
in
when
the
you say
organisms
concentrating, you don't filter it in that case. You actually freeze dry the entire sample.
Okay.
Now, if you're pretty sure, I mean, there are ways to filter it out, take the cells and test them, and take that supernatant and test just the supernatant, but unless you tell that lab this is what you want, they're going to process that whole sample and just tell you what's extracellular and intracellular. So the number you get back from that whole sample will be both. Okay? Does that answer your question?
Yeah. The new standards are based on extracellular concentrations.
So the what? Sorry.
The WHO standard, the 1 microgram per liter, is that based on extracellular or intracellular?
Well, if it's finished water, it's going to be mostly extracellular. You won't have any cells in there, hopefully. So it's just whatever's in the sample. And again, that guidance is drinking water, finished water. It's not the Bloom recreational water. There are separate guideline numbers for recreational. Those are more like 20 and 50. Does that answer the question? Yeah, but it's a good point. But you're right. Yeah, again, you shouldn't have any cells in finished water, so you're just measuring extracellular. Any other questions? Yep.
Based on your experience, if you do separate the intercellular from the extracellular, do you see any concentration trends? Like normally intra will be more higher?
Sure. Yeah. The intracellular should be much higher, but it depends on the toxins. Microcystin is mostly in the cells, so unless you've stressed those cells or the bloom is dying, 99% of it's going to be in the cell. With something like cylindrospermopsin, 80% of the toxin is outside of the cell. So you really have to process the whole thing. So that's why it's best, if you're doing toxin testing, you just process the whole thing. And you know, since that's where the exposure is, you're at least setting risk based on what's there. I don't know of a situation where you were more concerned about what's outside versus inside, if you're just wanting to know guidance.
The reason I'm asking is with a 5-day preservation time, some cells might die.
Mhm.
If they die, it seems that the toxins will be released.
Sure they will, yeah.
So if that is the case, right after your collection and analysis, you might have a lower concentration versus down the road you have higher concentration. If you do combine, that probably so.
Yeah, again, that's why combining, then you don't have those kind of what-if questions so much. Mhm. Yeah. Okay. This is Thai Lake in China. Some of the original, for reference, this is what the Ripley waters look like on the shoreline.